Okay, so we're going to talk a little bit about what should be in the lab report, in the osmosis lab report. We did this in class on Monday, but I did not record it, so this will be a nice reference for you for later. The first thing that I would direct your attention to is in the module, and you guys have to be able to see it kind of lightly behind me, I think. In the module 1, there is a document called the Biology Laboratory Guide. And if you happen to open that up, what the important part is here, where it says experimental lab reports. So it's on the beginning of the second page. And it tells you very specifically what must be included in each of the sections, as well as the rubric that I'm going to be using to grade your osmosis lab report. It tells exactly how much everything is worth and how I'm going to grade it. And I will have a copy of this with my handwriting on it and the points you earned stapled to your report or attached as a rubric to your report if you upload it. Okay, so this actually tells you pretty much everything that needs to be in the lab report, with a couple expansions that we talked about in class, right? So I'm going to go through it relatively quickly. So the first thing that has to be in your report is the first section is an introduction section. And as we noted in class on Monday, the introduction needs to include something about some description, definition of what osmosis is. Right? You can use your textbook, use your big college words, hypertonic, hypotonic, right? Concentration of water, diffusion across the membrane, all those things that we talked about. I would point out, and I did point out this out in class as well, that in the handout, osmosis is capitalized everywhere. It's not a proper noun. That's a secretarial error. And then she kindly made 150 copies of the handout for me. And like I said, I felt bad just throwing them in recycling. So I left it, right? So just be mindful of that. Osmosis is not a proper noun. Please only capitalize it when it's the beginning of the sentence. And the second part of your introduction needs to be a hypothesis. And in this case, it's hypotheses, most likely, because I would like you to tell me about what you think is going to happen in each of the beakers. I would like you to be very specific. I think in the 1% salt solution, I think in the 0% salt solution, the potatoes will gain weight, lose weight, right? Gain mass, lose weight, change total percent weight change, whatever words you want to use. But it would be best if you could tell me what you think is going to happen in all the beakers. If you want to lump the 2% and 3% beakers together, that's fine, because the same thing happened in those two beakers, right? For your materials and methods section, do not write out a list of all your materials and do not write out paragraphs telling me what you did. I was there, I saw it, I told you what to do, I watched you, you did what I told you to do, turned out fine. There is no hidden errors, there's no hidden human error. We're using electronic balances now. I mean, there's barely any errors of even reading the scales, right? It's fine. That's not what this is about. So for your materials and methods, I would like to see the following sentence. Please refer to the lab handout. That is worth 5 points. Do not write all that stuff out. I will give you 0 for that. Although I have a sneaking suspicion that a couple people will write it all out and then put that at the end, but they still get, it's just a waste of time. I want you to spend your time on the part of the lab report that's hard to do and that has the most meat in it. not like I gave my example in class, not spending an hour typing all that out while you're watching baseball or football and then being like, whew, I've worked on this lab report like all day. I'm going to finish that on Monday. No, you haven't done anything yet. You haven't gotten to the good part. Next section is your results section, and it must include 3 things. It must include a sample calculation this time, because believe it or not, I need to know that you can do the math. Because sometimes people can't, even though they're supposed to be through college algebra before they get into this class. So I need to see a sample calculation. You don't have to give me all of them. If you want to give me all of them, that's fine. Some people put them on their graph paper for the graph portion, that's fine. But there is a calculation for you to do at the bottom of the page. Final minus initial divided by final times 100, right? The weights. So your final weight, final mass, I should say, minus initial divided by initial times 100. Total percent weight change. I need to see that for at least one of your data points. That's all you need, basically, is the first number and the last number. And for some of the lab tables, when I went around after you guys were about finished, on occasion I said, don't use this for your initial, even though it's your first number. I circled like the second or third one down and said, use this for your initial number. I think I did that at your table on one of them, right? So I just wanted to mention that, because some people are, if they receive data from lab partners and they see that the initial is crossed off and the next one's circled, it's because I asked them to use that one as their initial number, right? The second thing in your results sections is the graph. And the graph, it's best if it's on graph paper, because that's easy. And the graph needs to have a title. It's not potato graph, or it's not osmosis graph. Titles in science papers describe what the graph is about, right? So it'll probably be a sentence. The axes need to be labeled, X and Y, and what they stand for. And then you have to actually plot your graph. Since the potatoes, some of them gained weight and some of them lost weight, we actually need 2 quadrants on our graph. We don't need these 2, even though they're technically there, right? But we need, on our y-axis, we need positive and negative numbers. So we need designations below 0 and above 0. The x-axis on graphs is always the independent variable. And the independent variable is the variable that you, as the scientist, experimenter, that you choose and or control, right? So in this case, what did you do differently in each of the beakers? What was different in each of the beakers? The amount of salt, right? So the percent salt solution then is the x-axis. So we're going to have 0, 1, 2, 3. I suggested to students in class, I would try to stretch that axis out as much as possible, because it's going to give you a more accurate number for your isotonic, what is potentially isotonic for this solution. So do not just put 0, first box 1, second box 2, third box 3 on your graph paper. Right? Stretch it out. In class, I did 0.2, or I would do that at least, or you could even do tenths. So I'm going to slide these down a little bit. So 0.2, 0.4, 0.6, 0.8, 1% salt, 1, 0.2, 0.4, 0.6, 0.8, 0.2% salt, 2, 0.4, 0.6, 0.8, 3% salt, right? Which leaves my y-axis to be the percent total weight change. And again, I would do it at least in 0.5% or more if you could.
Doing it in thirds is a little tricky because then it's harder to figure out between 0.6 and 0.9 or 0.3 and 0.6, right? It's just our brains don't work as easily that way, but it's up to you. Please make your scale going up equivalent to the scale you're using going down. So if you need to graph minus 9.6% for your 3% salt solution, I would go to 10, a positive 10 and a negative 10, because you never want the point on your graph to be the last mark, the last gradation on your scale. Okay, it's just it's a graphing thing. Don't use Excel because Excel doesn't want to make a graph like this. Excel wants to make you're going to ask to make a line graph and it's going to want to connect the dots, unless you know how to make Excel do this, which some of you may. If that's your thing, right? If that's your jam and you like to play with computers and figure out how to make Excel do what you want, knock yourself out. I think it's a lot less time consuming to just get out a piece of graph paper and make the graph. Okay. Your graph is going to look something like this. You're going to plot the numbers and you're going to make what we call best fit line. Do not connect the dots, right? It's not a perfect best fit line, excuse me, because we did not collect. What we should have done is I could have collected the data from every table in the class and we would have been able to make error bars, and then you could have actually made a best fit line, right? We didn't do that. So just take a ruler and get as close to as many of the points as you can. How about that? And then the third thing your results section needs is a sentence or two, some sentences describing the results, not explaining them. There is no because in this section. There is no I think in this section. It is just potatoes in the 0% salt solution gained 5.62% in weight. The potatoes in the 1% salt solution, blah, 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 blah. 2%, just a statement of the facts. In your discussion section, then you need to tell me why. And the discussion section for this lab report has two specific components. The first part is explain to me. Explain osmosis to me, I should have said. Explain osmosis to me with potatoes and salt solutions. All the gritty details. This is where you need to make it perfectly clear that you understand it well enough that when I read it, I can see that you understand it. And again, use all your college words. Which solutions were hypotonic? Why were they hypotonic? What was the water doing? What did that do to the weight of the potatoes? All right, explain it to me. I would advise after you do that, don't make it one long run-on sentence or three, right? After you write it out, because it'll be in your brain and you like, blah, 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 and you'll type it really fast, then read it out loud and see if it makes any sense and fix it, please, right? I would also remind you, we're comparing things across the membrane. So one side of the membrane has more water than the other side, not than the other side, right? You will be graded for grammar. The second thing that needs to be in your discussion is what % salt is isotonic for your potatoes. And the answer to that question is where your line crosses X. We are doing an interpretation or an extrapolation from the data we collected. Our best guess, this is actually isotonic, whatever that number is. That's why I said stretch this axis out, because you're going to get a more accurate count on that number, right? If this is 1% and this is 1.2% and it falls right in between there, I want you to say our potato had 1.1% salt in it, right, from the graph. sometimes people say our potato had 6% salt. I'm like, where does it say that on your graph? Right? So use the graph to give me the answer to that question, right? According to figure 1, salt in our potato was 1.1%. Done. Your conclusion is super simple as well. Do your results agree or disagree with your hypothesis? And as I told everyone in class on Monday, I don't care if you had the right hypothesis, if you didn't have, you're not going to get more points if they didn't match or if they do match. That's not ideally, as your lab partner pointed out, Zach pointed out, we didn't make the hypothesis before we started. Just pretend, you know, ideally, yes, you would have made the hypothesis first, then weighed the potatoes, and then been like, oh, what we thought was going to happen didn't happen, or whatever. One last thing, the water is what is moving, not the salt. Don't tell me the salt goes in and out of the potatoes, because whoosh, with the red pen, we didn't measure the salt. We didn't measure the rate. Don't tell me we measured the rate of water moving in and out of the potatoes. We did not. Rate is a specific mathematical calculation of an event over a particular period of time. And you would have to calculate that. We did not calculate a rate. We calculated percent weight change. So be careful. I want you to use as much of the scientific language as you can, but don't use language that doesn't make sense or isn't true for this particular experiment. I'm glad we did that because I should have done that last week.